Hello, my name is Carlos Brian Francia, and welcome back to another lecture video. Today we're going to be learning about the immune system and what makes up the immune system, how the immune system defeats pathogens and bacteria and viruses. But before we begin the discussion of B cells and T cells, we have to discuss the anatomy of the lymphatic system. That's essentially the highway that the B cells and T cells and macrophages, they use the lymphatic system to travel throughout the body. So let's discuss the organs and the highway system that the immune system uses. Within the tissues, there's an area between the arterioles and the venules, and this is the capillary. So between the capillaries, you will have the lymphatic vessels. And the lymphatic vessels, all they do is that they transport the immune cells. For instance, white blood cells, macrophages, B cells, T cells. And the way that these lymphatic vessels are formed is that they're made up of endothelial cells. So they're very thin. These little cells are just one cell thick. And they are tethered to the tissue with these little anchors. So these guys are little anchors, and they're called collagen fibers. When the solute concentration is low inside the tissues, the collagen fibers are going to prevent the fluid from the lymphatic vessel from exiting. So we don't want this fluid inside this lymphatic vessel to exit. So when the solute concentration is low inside the tissue, instead of the fluid leaving the lymphatic vessel, we close it. So the collagen fiber is going to close the endothelial cells because these guys are kind of like little doors. So we don't want that to happen. But what happens if the solute is increased inside the tissue? So if there's a lot of cells, maybe there's some items inside the tissue, and the solute is increased, well, instead of being closed, the collagen is going to open the endothelial flaps. And so now we have all the solute, maybe it's fluid, and it's going to enter the lymphatic vessel. And when the fluid from the interstitial space enters the lymphatic vessel, that becomes lymph. So lymph is just interstitial fluid. So this fluid that may be here is going to go inside the lymphatic system, and this interstitial fluid is called lymph. Sometimes there's a failure of the collagen fibers. So the collagen fibers prevent the lymph from exiting the lymphatic vessels because it keeps the little flaps closed, the little endothelial flaps. But if the collagen fiber doesn't do its job, the lymph fluid is going to leave and build up in the interstitial space, and that causes lymphedema. So that is just edema caused by the lymph. And lymph is just the interstitial fluid inside the lymphatic vessel. Here you'll notice that there's actually a valve. And these guys are one-way valves. These are one-way valves. And they're called semilunar valves. As the lymph travels, let's say it travels downward from this picture, it will not be able to go upwards because these semilunar valves prevent any backflow. That prevents any of the lymph fluid from going back from what it started from. And you'll notice that there is no smooth muscle here. So for instance, in the arterioles and in the venules, they have little smooth muscles on the side, and that helps them to contract and to relax. But that's not present inside lymphatic vessels. So you're probably wondering, well, how does that lymph fluid travel in the lymphatic system? Well, the way that it travels is that it uses your body. What do I mean by that? When you're moving, when you're breathing, when you're exercising, your skeletal muscles squeeze the lymphatic vessel. And so the muscles right here, let's say that this is in your arm, the arm muscles will squeeze this portion and squeeze that portion, and that's going to force the lymphatic fluid, the lymph fluid, to travel. So it uses your movements to flow through your body. It doesn't use smooth muscles. So medications that lower your blood pressure by relaxing the smooth muscles, that's not going to do anything to the lymphatic vessels because they don't have smooth muscles. They're really thin. They only have one cell of endothelial cells, so they don't have any smooth muscles. And when you breathe, when you move, when you exercise, you're pushing that lymphatic fluid into the lymphatic circulation. But what uses the lymphatic circulatory system? Well, the lymphatic system can transport certain vitamins. For instance, fat-soluble vitamins are vitamin K, A, D, and E. These guys are fat-soluble. And an easy way to remember them is to just put CADE. That's a name. We can also transport lipids. When you eat a fatty meal, let's say you just had ribs, the lipids are not going to enter the arterial, they're not going to enter the venules, right? They're not going to enter the systemic circulatory system. You don't want any fats in your blood. Instead, they're going to enter the lymphatic system. And the lymphatic system have lacteals. So lacteals are just little vessels inside the villi, inside the small intestines. So as the lipids are processed in the small intestines, they enter the lacteals, and now the lipids will be transferred to the liver, they're filtered, and then the liver shoots the nutrients into the circulatory system. When we have an infection, let's say we have an infection here in the interstitial space, the B cells and T cells will enter the lymphatic system, and then they're going to leave and enter the interstitial space, and they're going to fight the pathogen. After the pathogen is destroyed, we have little bits and pieces of pathogen. We have a cell called macrophages, and these macrophages are going to eat the little bits and pieces, and they're going to get rid of the trash, and they also use the lymphatic system, and they enter into the interstitial space and they do their jobs. So the lymphatic system is kind of like a special highway for our reinforcements, for the military, for the garbage disposal. We can transport nutrients, we can transport vitamins, we can transport lipids, we can transport T cells, B cells, macrophages, all those great things, and we can rush to an area that is being attacked by pathogens, and we use the lymphatic system. But now we have to describe how we get items from the lymphatic system and push them into the circulatory system. So how do we go from the lymphatic system into the major circulatory system with the blood? Here we have the lymphatic system. And the lymphatic system kind of looks like the circulatory system because it's found throughout the entire body. Now, in the circulatory system, the arterioles become capillaries, and the capillaries become venules. But in the lymphatic system, we have something called the lymphatic capillaries, which you just saw in the previous slide. And these guys are going to thicken up, and they become something called ducts. They're called ducts. In the body, we have two types of ducts. On the right side of the body, we have the lymphatic duct. On the left side of the body, we have something called the thoracic duct. And this thoracic duct is a lot thicker than the right lymphatic duct. The right lymphatic duct is very thin, and the thoracic duct of the left side of the body is thicker. So sometimes we have to ask ourselves, why is the thoracic duct thicker? Well, it's on the left side of the heart, and the left side of the heart has more muscle because it has to shoot the blood further into the body. So we can kind of use that logic for the lymphatic system. Since we're on the left side of the heart, we have to be thicker because there are thicker arteries, there are thicker veins, so there's more fluid coming in. Therefore, we have to have a thicker duct to accommodate that fluid. We have to have a larger duct to accommodate this amount of fluid. And so we're going to call ourselves the thoracic duct. What does thoracic mean? Thoracic just means that we're on the upper part of the body. And in fact, the right lymphatic duct is going to take fluid, lymph fluid, from the upper part of the right side of the body. So for instance, the right arm, the right shoulder, the right side of the head, that is going to go into the right lymphatic duct. For the left portion of the body, we're going to be taking fluid from the left portion of the head, the left shoulder, and the left arm, and the left chest. and we're going to put that into the thoracic duct. From then on, the thoracic duct goes into the subclavian vein. What is the subclavian vein? The subclavian vein essentially are these little veins right here, right underneath the clavicles. And so for the left portion of the body, we have the thoracic duct, we're going to call that TD, and it goes into the subclavian vein, the left subclavian vein. For the right portion of the body, we have the lymphatic duct, and it goes into the right subclavian vein. So basically, the right side of the body has the lymphatic duct, which is smaller. It goes into the right subclavian vein. For the left portion of the body, we have a thicker duct called the thoracic duct, and that just goes from the thoracic duct into the left subclavian vein. But we just talked about the upper portion of the body. What about the lower portion? What about the legs and the pelvis? What about this area? Well, the lymph fluid is going to accumulate inside this area called the cisterna chyli. And the cisterna chyli kind of looks like a little pocket. And this pocket will swell up with lymph fluid from the lower body, and then it's going to push this lymph fluid into this thoracic duct. So the cisterna chyli goes into the left thoracic duct. And notice there's a lot of lymph fluid being processed inside the cisterna chyli. This takes the entire lymph fluid from the lower portion of the body, and then it shoots it into the thoracic duct. That's why the thoracic duct is so thick, because it takes in not only fluid from the left portion of the body, but it also takes in fluid from the lower portion of the body. The lower portion of the body goes into the cisterna chyli, and the cisterna chyli pushes that lymphatic fluid into the thoracic duct. So that's why the thoracic duct is so thick. It's so much wider than the right lymphatic duct. After we go from the lymphatic ducts, or the thoracic duct, into the subclavian veins will eventually reach the heart, and the heart will go into the systemic circulatory system. So if I had a lipid, or if I had vitamins K, A, D, and E, they would go from the thoracic duct or the lymphatic duct into the subclavian vein, and from the vein they go into the heart, and the heart will shoot the lipids, they will shoot the vitamins into the systemic circulatory system. So now you have these lipids and vitamins going into the tissues, going into the organs via blood. So this is the transfer between the lymphatic system into the circulatory system. Now that we've discussed the lymphatic system, we have to talk about our main characters of the immune system. We talked about the highways that they use, where we gotta talk about the people that use it. So here, we're gonna be focusing on these guys. So these guys are called the leukocytes. So these are leukocytes. And these guys right here are also called leukocytes, but they're a little special because these guys live inside the lymph organs. They live inside lymphatic organs. So for instance, the lymph nodes, or the spleen, or even the bone marrow or the thymus. These guys live in that area. And because they live in this area, we call them lymphocytes. So lymphocytes are a special subdivision of leukocytes. And leukocytes, specifically leuko, that just means white. Though when they were being discovered in the early 1900s, people saw that they were white and they called them leukocytes. That is the white blood cell. And these guys are white blood cells. These guys are also white blood cells, but specifically they live in the lymphatic system. So they're going to be called lymphocytes. We have another way of dividing lymphocytes and leukocytes, and they are called granular or agranular. Granular means that you have granules inside the cytoplasm. But what is that? Well, for instance, neutrophils, they are going to have something inside their cytoplasm called granules, and when they are interacting with a pathogen, they will release those little granules. It kind of looks like dust, and they will release this dust onto the pathogen, and that pathogen will die. Sometimes the cells will release their granules, and these granules are going to have different messages. So they might signal for another immune cell to do something, and those are granules. They kind of look like little freckles inside the cell. So neutrophils have those little freckles. Basophils have those freckles. Eosinophils also have these freckles. And these guys, again, are called leukocytes. But a certain lymphocyte also has granules. And that lymphocyte is called the natural killer cell. So in immunology, natural killer cells are often abbreviated as NK, natural killer cells. And these guys have those granules, and those granules have something called perforins and granozymes. And we'll talk about that later, but those guys are going to release it onto the pathogen, and the pathogen dies when the enzymes from the granules interact with the pathogen. Likewise, we can talk about cells that don't have those granules. Instead, they're going to interact with pathogens in a different way. But T cells, and B cells, and also something called a monocyte, those guys do not have granules. So monocytes, these guys do not have granules. Instead, they're going to be having different ways of killing pathogens. Specifically, if we talk about monocytes, they can evolve, or they can differentiate into different things. So the monocyte, which is agranular, can become either a macrophage. So these guys right here, they're gonna nibble on the pathogens. They don't shoot anything, they just nibble on them and they eat them via phagocytosis. Or the monocyte can be something called a dendritic cell. And dendritic cells can also nibble on pathogens. They can phagocytize the pathogens, but these guys have a lot of arms. They kind of look like little octopuses inside the body. And since they have so many arms, they can take a lot of pathogens and phagocytize them into the dendritic cell. So these guys don't have any granules, but they are derived from the monocyte. And the monocyte is a white blood cell. So these guys are also called white blood cells. And so there are always going to be exceptions to the rules. For instance, you can't say, oh, all lymphocytes are agranular. That's not true, because a natural killer cell is a lymphocyte, but it has grains. You can't say all leukocytes are granular. That's not true, because the monocyte is a leukocyte. However, it is agranular. So the exceptions are the NK, which is a lymphocyte that is actually granular, and the monocyte, which is agranular, but is also a leukocyte. Now let's talk about the celebrities of the immune system. And they are called T lymphocytes and B lymphocytes. Another way to call them are T cells and B cells. And they help us survive every day. But where are they made? T cells and B cells are made inside the bone marrow. And right here is the bone marrow. Notice that we have something that looks yellow. This is the yellow marrow. And that's just a large collection of fat inside the bone. But really, we're interested in this red portion. This red portion will create T cells, and it's also going to create B cells. When B cells are young, they're going to stay inside the bone marrow, and they're going to fully develop. They age, they become mature. So B stands for bone. The B cells will remain inside the bone marrow. The T cells, however, are a little different. The T cells are made inside the bone marrow, but when they are young, they're going to leave the bone marrow and they're going to travel into another organ called the thymus. And the thymus is located within your neck. So the T cells are going to leave the bone marrow, and then they will go into the thymus to mature. They will learn how to fight for the immune system. They are mature inside the thymus. So both of these guys are going to originate from the bone marrow. The B cells stay inside the bone marrow until they are needed, but the T cells leave the bone marrow and go into the thymus so that they can mature and learn how to fight. The B cells learn how to fight inside the bone marrow. But what do the lymphocytes do? Well, when we look at the natural killer cell, the natural killer cell has granules that can kill friendly cells inside the body. Why do we want to kill our own cells? Well, let's say that we have a cell right here, and this guy is friendly. So this guy is one of us. But this guy has little viruses inside, and it is infected. And if the virus keeps replicating, eventually it will fill the friendly cell up, and the cell will explode. And now the virus is going to go all over the body and continue infecting other cells until the host dies. And so we have natural killer cells that have the little granules, and the natural killer cell is going to sense that something is wrong with this friendly cell, and it's going to poke holes, and it's going to kill this friendly cell. So we have to kill the factory before the virus is replicated and explodes. So that's what the natural killer cell does. It kills the friendly cells inside the body that are infected with a virus or a pathogen or a bacteria. So this doesn't have to be a virus. It could be a pathogen. It can be a bacteria. It could be a parasite. It just has to live inside of the friendly cell. And the natural killer cell is going to kill this guy right here, and it's going to prevent that replication of the proteins. When we look at T cells, we have two different types of T cells. We have the helper T cells, and then we also have something called cytotoxic T cells. And we're going to talk about them more in the future, but for right now, you just got to know that helper T cells help the immune system. They release messages into the body, and those messages can interact with neutrophils, they can interact with B cells, they can interact with a lot of immune cells, and they help those cells. How do they help them? Well, a helper T cell might release a message and it says, hey, there's a guy right here that has a virus and he's in the left arm. So now all the neutrophils and the macrophages and the B cells, they're going to rush to the left arm, and then they're going to do their thing. They're going to eliminate this guy. They're going to help out the NK cells. So the helper T cells release messages. And they also increase the immune response, or they can decrease the immune response. So it really depends on what the T cell wants to do. But what about cytotoxic T cells? Cytotoxic T cells are actually going to eliminate pathogens. They're going to eliminate things that hurt us. So this T cell that is cytotoxic, let's call it a CT, cytotoxic T cell. This guy right here, he's going to go up to this friendly cell, and this friendly cell has a virus, and it says, hey, you have a virus, I have to eliminate you, and it's going to interact with this cell, and it's going to eliminate it. It's going to kill the infected cell. So cytotoxic T cells also work with natural killer cells. So natural killer cells and cytotoxic cells are a part of the immune response that will eliminate the pathogens. They will kill the pathogen or the infected cells with viruses. Here we have the B lymphocytes, and the B lymphocytes are going to make something special called antibodies. Antibodies. The macrophage is just going to be wandering around the body, and it sees, let's say, it sees a pathogen. It has this pathogen, and this one right here is a bacteria. So this guy right here is a bacteria, and the macrophage is going to nibble on the bacteria, and it's going to take a part of this bacteria and present it to the B cell. The B cell is then going to make antibodies, and these antibodies kind of look like little Y shapes. They look like little Y shapes right there. And these little Y shapes are going to stick to the bacteria. So we're going to make it like this. And then a process happens, and it's a little complicated, but this process allows a hole to be formed inside the bacteria, and water rushes into the bacteria, and it explodes and it dies. So this bacteria is going to be covered by antibodies created by the B cell, and that is very specific. So this is actually part of the specific immune response. There's something called the specific immune response. It's also known as the adaptive immune response, and there's something called the non-specific immune response, also known as the innate immune response. We'll talk about that in a few minutes, but right now we're looking at the B cells. When the B cell takes in the bacteria item from the macrophage, it will release antibodies. But before it can release antibodies, it becomes something called a plasma cell. And plasma cells are basically machines that will create a large amount of antibodies. It's something extreme, like 2,000 antibodies a second. So it becomes a plasma cells, and it machine gun shoots antibodies at the pathogen. It also makes something called a memory cell. So this is a memory cell. And essentially it says, hey, I'm going to remember that we fought this bacteria. So if that bacteria comes again, maybe in a week or in 10 years or 20 years, I know how to deal with it. I'm not going to forget how we killed this bacteria. I'm not going to forget how we eliminated this virus. I'm not going to forget how we eliminated this worm or parasite. I'm going to remember. So if we ever see this pathogen again, this memory cell is going to create a large amount of plasma cells. And these plasma cells have the antibodies that are specific to attach to this pathogen. And then when the antibodies attach to this pathogen, a whole process happens that creates little holes inside the pathogen, and water rushes in and it kills that pathogen. So the memory cells remember how we dealt with it, and the antibodies act on it. So another way that you can call plasma cells are effector cells, because they have an effect on the pathogen. However, we don't really call them effector cells. Scientists don't really like that name for some reason. They would rather have you call it plasma cells. So plasma cells make antibodies, and memory cells remember how we dealt with those pathogens. Let's talk about the thymus. The thymus is an organ that is part of the immune system. And the thymus is where the T cells that are immature go, and they learn how to fight for the immune system. The thymus has an exterior that is a little firm, and this exterior is called the fibrous capsule. So this capsule encapsulates or surrounds the thymus. Inside the thymus, we have two sections. We have something called the cortex and something called the medulla. And it's very similar to the kidneys. The kidneys have an outer part called the cortex and an inner part called the medulla. If you look at this slide, you'll notice that the cortex is a lot darker than the medulla. And the reason why is because there's a lot more cells in here. It's very condensed and compact with cells. However, in the medulla, there's a little bit of cells, and they're further apart, so they're not as concentrated. And therefore, the dye that is used to look at the specimen is not concentrated inside the medulla. As we learned previously, the T cells begin their development inside the bone marrow, and then they will leave the bone marrow and enter the thymus. But they are young, they are immature, and they're called something like thymocytes. So the thymocytes are just immature T cells, and they don't know how to fight against pathogens. So they leave the bone marrow and they enter the thymus, and they learn how to fight the pathogens. After they graduate from Thymus University, they become T cells. So let's actually look at how the T cells learn how to fight pathogens, and how do they learn not to fight the own friendly cells. How do we know not to attack the body and only to attack the pathogens? The thymocyte, which is just a immature T cell, enters the outer portion of the thymus, and that is called the cortex. And now the thymocyte attends Thymus University, and that's where it's going to learn how to fight pathogens. How do we fight them? Well, first we have to take classes, and these classes are being held by a professor called the thymic epithelial cell. And this professor looks at the thymocyte and it says, hey, I gotta make sure you know how to read, because in your job in the immune system, you're gonna be reading a lot. You're going to be reading all day, every day. So make sure you know how to read. And so the thymic epithelial cell is going to hand the thymocyte a test. It looks like a little piece of paper. And the thymocyte looks at it and it says, what is this? And the piece of paper is called the major histocompatibility complex. And a lot of immunologists will take this big word and they're going to put it as MHC. So that is the major histocompatibility complex. The major histocompatibility complex is essentially just a piece of paper that has words on it. And these words say something like there's a E. coli infection or a mumps infection, or there's a worm inside the small intestine. And we got to make sure that the thymocyte is able to read it. We will undergo positive selection. We will select for thymocytes that are able to read the major histocompatibility complex. Can you read this piece of paper? Well, this guy right here, this thymocyte, he can read this piece of paper, and so he is allowed to stay in Thymus University. However, this thymocyte cannot read the major histocompatibility complex. It can't read this piece of paper. And if you can't read, then most likely you're not going to go to university. You're going to be kicked out. And unfortunately for this thymocyte, they are killed by the by the thymus, and so they are kicked out of Thymus University. After the thymocyte completes the first class, it's going to get two types of tools. It's going to get something called the CD4 receptor, and it gets something called the CD8 receptor. After the thymocyte graduates from Thymus University, it will either keep one of the receptors. It will either keep a CD4 receptor or it will keep the CD8 receptor. Right now, the thymocyte has both. So it has both of these guys right now, because in university, you don't know what you're going to do. You don't know if you're going to do an English major or a chemistry major or a biology major. And so when you're in that university, you have different classes. You have an English class, a chemistry class, or a biology class. And you say, hey, I really liked chemistry. I loved that class. I might major in chemistry. It's the same thing for thymocytes. They don't know what they're going to become. They might major in CD4 or they might major in CD8. But right now they're experimenting and they have both. And because they have both the CD4 receptors or CD8 receptors, they're called double positive. When we first started our classes at Thymus University, we didn't have CD4 receptors. We didn't have CD8 receptors. And so we are called double negative. We have negative, negative, double negative. At the end, you're only going to have one or the other. And so you will have a positive and negative. So if I graduate from Thymus University, I can become a CD4 T cell. I am positive for the CD4 receptor, but I do not have the CD8 receptor. And so I am only a CD4 T cell. After we just graduated from the positive selection course, we have to go to the negative selection course. We will have another professor, and that professor is called the dendritic cell. And what the dendritic cell does is that it's going to get a piece of paper, it's going to get the major histocompatibility complex, and it will present it to the T cell. And on this little piece of paper, on the major histocompatibility complex, are antigens. Antigens are proteins. So the dendritic cell is going to write something on this piece of paper and it says kidney, or liver, or self. On this piece of paper, the antigens are written. They are presented onto the major histocompatibility complex. These antigens are healthy. So if the thymocyte says, okay, I can read this piece of paper, I want to attack the kidney, I want to attack the liver, and I want to attack self. I'm going to attack it, I'm going to use my CD4, I'm going to use my CD8. Well, you failed. Because look, the dendritic cell is trying to say, hey, you better not attack the person's body because there's nothing wrong with these cells. If you choose to bind to these healthy cells, you're going to be kicked out of the class. And so we want to select for T cells, for thymocytes that say, hey, I don't want to attack the kidney. I don't want to attack the liver. I don't want to attack this person's body. So that is called negative selection. And that's different from positive selection, because positive selection, they will pass you, they will allow you to graduate if you're able to read the piece of paper. In negative selection, they are going to allow you to graduate if you say, no, I don't want to attack the kidney, I don't want to attack the liver, I don't want to attack self. If you choose to attack self, you're going to be kicked out of the course, and that is what happens here. So in this picture right there, the thymocyte looks at the major histocompatibility complex and it says, hey, there's an antigen, this antigen is healthy, let's attack it. I'm going to attack Brian's body. Well, you get kicked out of the course. For this portion right here, the thymocyte says, hey, I can read the major histocompatibility complex. I can recognize that this antigen is healthy. It's part of the kidney, and there's nothing wrong with the kidney. I'm not going to do anything to it. I'm going to allow it to live. And that allows the thymocyte to graduate the class, and it's able to leave the medulla of the thymus. So now we are fully graduated. And when you graduate, the thymocyte says, you know, during my courses, I really liked being a CD4 cell, and therefore I'm going to keep the CD4 receptor. And this is known as a CD4 cell, also known as a helper cell. I liked helping people. I'm going to stay being a helper T cell. Or it might have graduated from Thymus University and said, I didn't really like being a CD4 cell. I kind of liked being a CD8. I liked attacking things. And this guy is going to be called a cytotoxic T cell. So you can see that the thymocyte is going to lose one receptor and keep the other receptor. It can keep the CD4 receptor, but in doing so, it's going to lose the CD8. Or it can keep the CD8 receptor and it will lose the CD4 receptor. And this is permanent. You can't just switch your major in Thymus University. So now we have the mature cytotoxic T cell and the mature T helper cell. When we're talking about immunology, immunologists will write T helper cells like this, TH, or T helper. And they will write cytotoxic cells like this, TC, T cell that is cytotoxic. From then on, the mature T cells are going to leave the thymus and they're going to enter the secondary lymphatic organs. What is a lymphatic organ? Well, there are two types of lymphatic organs. There's the primary, and the primary, that's just the thymus, and also the bone marrow. The secondary, that is the spleen, lymph nodes, and really a lymph node is like this little pocket right here. Here's a lymph node right there. Here's another one and another one. They're just kind of like little centers where the lymphatic vessels congregate. They all join at the lymph nodes. And then we have a smaller lymph node, and that's specialized, and we call them lymph nodules. And a lymph nodule are your tonsils or your adenoids. So that's the adenoid in the back of the throat. And then we have your little tonsils right next to the tongue. These are lymph nodules, and they have a large concentration of T cells and B cells. So when the pathogens enter from the mouth, the tonsils are going to sense the pathogens and they're going to release T cells to kind of take away the pathogen. So the secondary lymphatic organs are the spleen for the blood, the lymph nodes right here in green, and the lymph nodules, which are little tonsils in your body. I'm kind of interested in seeing what a lymph node looks like. What does this look like right here? So let's zoom in and see a cross section of a lymph node and what it looks like and what it does. This is the lymph node. This is the lymph node. And the lymph node is going to filter the lymphatic fluid. When the lymphatic fluid enters into the afferent vessels, afferent means entering, it will exit the lymph node. Efferent means exit. E is for exit. A is for arriving, afferent. When the lymph enters the lymph nodes, sometimes it has bacteria. So we can have little bacteria hanging out here, or there's a pathogen that was picked up by the lymph fluid, and now the bacteria is inside the lymph node. Well, what else is inside the lymph node? There's T cells and B cells and macrophages and dendritic cells, and when they see an intruder, they will kill the intruder inside the lymph node, and also they're going to start multiplying. So now we have the immune response occurring. So the lymph nodes are going to start creating more immune cells, and these immune cells are going to leave, and they're going to go into the systemic circulatory system, and they're going to travel throughout the body and try to find more pathogens to eliminate. So this guy right here is kind of like HQ, where all the immune cells they kind of relax and they hang out. And when they relax and hang out, you can think of it as a club. This is club lymph node, node. When the lymphatic fluid enters, it enters at the subcapsular sinus because the lymph node is made from a capsule, much like the thymus. But the subcapsular sinus is like the door. So this right here is the door. And now you have your little lymph fluid right there. You see? And these guys are going to be the macrophages. And we have a dendritic cell right there. Dendritic. So these guys are waiting to go into club node, and they're the lymph fluid, but then they have to pass through macrophages. They have to pass through dendritic cells that are really concentrated inside the subcapsular sinus. But notice how this person right here kind of looks mean, and she has like a bad attitude or something. Let's call her a pathogen. And I'm sorry if she's watching this video, but she's a pathogen. So she's trying to sneak her way into the lymph node. But this guy sees her and he says, we're not going to let you pass. And in fact, we're going to alert security. And now the security guards inside the lymph node are going to start multiplying, and they're going to exit the lymph node to find more people like her. So they want to find more pathogens. But she is dealt with inside the lymph node. So she's not going to be able to to have her party. So in the subcapsular sinus, we have macrophages and dendritic cells, and they're going to be screening the lymph fluid. So sometimes the lymph fluid comes in, they have vitamins and nutrients, and that's cool. Sometimes there's T cells, sometimes there's a B cell, and they're going to be able to go into the lymph node, but the pathogen is going to be destroyed inside the lymph node. And now you have security guards that will leave the lymph node, and they're going to go to the other parts of the body to find more pathogens to fight. Inside the lymph node, near the cortex, we have these little centers, and these centers are called germinal centers. So now we're inside the club, and you notice that there's a large collection of people in a circle, and that is called a mosh pit. And this mosh pit has a bunch of B cells. So these B cells are going to be inside the germinal centers, and they are rapidly dividing. So these guys are very rapidly dividing. So let's say that there was a pathogen. Well, now the B cells are going to start multiplying, and they multiply inside the mosh pit that is called the germinal centers. Now, around this germinal center, around this mosh pit, are the security guards. And you know who the security guards are? The security guards are actually T cells. And you also have some macrophages. So these macrophages and dendritic cells and T cells, they surround the germinal centers and they protect the B cells. In addition to this, you can see that the security guard is talking to a walkie-talkie, and they're sending more signals to say, hey, we found a pathogen outside, she has a bad attitude, there might be more inside the body, we're about to leave lymph node number 2, and we're going to meet you in the right leg. So these guys are talking to themselves, and these T cells are also going to be leaving the body. So they signal for more T cell creation, and they also signal for more B cell creation. So this occurs within the germinal center. The germinal center is where the B cells multiply, and around the germinal centers are the T cells and the macrophages and dendritic cells, and they're all getting ready to leave the lymph node because there was a pathogen. And when the immune response leaves the lymph node, it will leave via the efferent lymphatic vessels. Around the germinal centers are called trabeculae. In trabeculae, they just keep one mosh pit separated from the other mosh pit. They just keep one geminal center away from the other geminal center. So we have kind of like these little areas. So you might have mosh pit number 1, mosh pit number 2, just different areas inside the lymph node. The next lymphatic organ, the secondary lymphatic organ, is called the spleen. And the spleen, all that does is that it's going to filter the blood, and it's going to monitor the contents of the blood. So the spleen doesn't really have a hard exterior surface. It does not have a capsule. And therefore, if you punch somebody really hard and really calculated, you can rupture the spleen, and that causes major blood loss, and it's not good for the person. So it's very fragile. It's a very fragile organ. And if you do a dissection on a person, the spleen is super red. Why is it red? Well, notice that we have the arterial and the venule. All the blood eventually passes into the spleen, and when it passes into the spleen, it will contact two areas. So notice that there is something called the red pulp, and there's something called the white pulp. The red pulp has a high concentration of macrophages. And that filters the blood. So for instance, our red blood cells, sometimes they get old and they have to be recycled. So if we see any old red blood cells right here, they get eaten by the macrophages, and then they are recycled by the body to make new red blood cells. However, let's say that we have a pathogen. Let's say that we have E. coli, and E. coli is found inside the arteriole. Well, now the white pulp comes into action. What is the white pulp? The white pulp has a high concentration of immune cells. So it also has these leukocytes, for instance, T cells and B cells, and some other immune cells, but these are the main two. And they notice that there is E. coli. So first, the dendritic cells right here are going to nibble on the E. coli, and they're going to present the E. coli antigen, which is like a little protein. They present it to the B cells, they present it to the T cells, and now the T cells and B cells, they say, hey, there's something wrong with the blood. We have to multiply and do an immune response. So the white pulp is basically the immune system alarm for the blood. And the macrophages inside the red pulp, that's going to be the filter. So the red pulp is the filter for the blood, and the white pulp is the alarm system for the blood. That senses that there's a pathogen inside the blood, and we have to do an immediate response. Here we have the histology of the spleen. That's how the spleen looks like in a microscope. And notice that we have the red pulp, and on here we have the white pulp. So this is just an area of a high concentration of lymphocytes, of T cells and B cells, and we have the germinal center. But between the red pulp and the white pulp, we have something right here, and that is called the marginal zone. And basically, the marginal zone, well, highlighted right there, is where the macrophages are nibbling on the pathogen, let's say E. coli. The macrophages are taking the E. coli antigens and they put it into the marginal zone. So here we have the little antigens kind of floating around in a high concentration. And now the T cells and the B cells are going to take these little antigens and they're going to start multiplying the T cells and B cells. So that's how we make the immune response. So if you ever see on the exam, what is a marginal zone? A marginal zone, in terms of the spleen, is the area between the red pulp and the white pulp, and it has a high concentration of antigens. So after the E. coli bacteria is killed, it has little bits and pieces left over. Those are going to be the antigens, and they're going to be presented into the B cells and T cells of the white pulp. Now let's talk about the lymph nodules. Lymph nodules are basically smaller lymph nodes, and these lymph nodules have no capsules at all. A good example of lymph nodules are your tonsils. Here in the mouth, we have something called the palatine tonsils. And these palatine tonsils, they allow us to kill pathogens in our food. For instance, let's say that I'm eating from a buffet, And the food that I'm eating, unfortunately, has E. coli. So now we have E. coli. And the E. coli enters the mouth right here and is going to be attracted to these tonsils. So now we have E. coli kind of congregating inside the tonsils. If we look at the tonsils in detail, we'll notice that there's something called a crypt. And a crypt looks like a long hallway, a very long hallway. And E. coli is very curious. So E. coli is going to wander inside the crypt and is going to enter further and further and further down the tonsils. Well, unfortunately for the E. coli, this crypt has a high concentration of lymphocytes. So these lymphocytes are going to attack the E. coli. We have the T cells, we have the B cells, and then we have some other immune responses. But they're going to use the crypt as a trap. So these crypts are very large invaginations inside the tonsils. A lot of scientists believe that we have tonsils as children to kind of train our T cells to respond to pathogens. So there's a high concentration of T cells right here, and they're training. They just graduated from Thymus University, but they've never seen a pathogen. And so when you eat food, this food has bacteria, and so the T cells learn how to deal with that bacteria. These foods might have parasites or maybe even viruses, but now the T cells are learning how to deal with the viruses, they learn how to deal with the parasites, and they learn it inside the lymph nodules. So what do we learn about lymph nodules? Well, lymph nodules have no capsules, they're very soft, they have a high concentration of T cells, they have crypts, and the crypts are traps for bacteria and pathogens, and when they reach further into the crypt, T cells and B cells come out and they attack the pathogens. Here we have something called the malt. So the entire thing that you see here is called mucosal associated lymphoid tissue. And an acronym for this is MALT. So the MALT is often seen within the small intestines, the breast tissue, and the eyes. So for instance, whenever you're eating food and you eat something that has a pathogen in it, like let's say you're eating salad and that salad has E. coli, the Peyer's patches or the MALT is going to kind of look at the pathogens inside of the food. And specifically, when we look at the small intestines, we're going to notice that we have geminal centers, and the geminal centers themselves are called Peyer's patches, and we have a high concentration of Peyer's patches within the small intestines. And the Peyer's patches, they're going to have something called microfolds, the microfolds, also known as M cells. So little bits and pieces of food are going to be passing through the small intestines, and the microfolds of the Peyer's patches are going to be looking at the pathogens inside of the food. So let's say that we have E. coli inside of that food. Well, now the B cells and the T cells are going to start multiplying, and they're going to mount an immune response. So that prevents the person from getting sick when they're eating. We don't want to get overly sick when we eat something that has E. coli. And if we do eat something that has E. coli, we want the body to fight it. We also have something called BALT. And BALT is the same thing as malt, except that it's located inside the bronchi. So that's located in the lungs. Whenever we breathe in air, we breathe in bacteria. And the BALT tissue prevents that bacteria from causing any further damage. So that helps us regulate the bacteria inside our respiratory system. We don't want to breathe in bacteria, and if we do breathe in bacteria, we want to stop it. And so these guys right here, the malt tissue and the BALT tissue, help us limit the bacteria inside the body. Now that we've discussed the anatomy of the immune system, let's talk about the two different pathways for the immune system. There's the innate immune system, and then there's the adaptive immune system. The innate immune system is non-specific, while the adaptive immune system is called specific. But what do I mean by specific and non-specific? Non-specific means that's the body's way of preventing pathogens or really anything from entering the body. It doesn't matter if it's a piece of dirt or dust or if it's E. coli or tuberculosis, it wants to keep foreign items out of the body. So this one doesn't really care what it's keeping out. It could keep out dust, it could keep out bacteria, it could keep out a virus. It is non-specific. And whenever we're dealing with something, we have two steps. The first step is going to be non-specific, also known as the innate immunity. And the second step is going to be specific. That is called the adaptive immunity. But right now, let's talk about the non-specific immunity. Part of the non-specific immunity is called the barrier. So the barrier system. What is the barrier system? The barrier system, it is essentially skin, hair, mucus, etc. For instance, when we look at the nose, we notice that there is skin on the nose. The bacteria want to enter the body. However, the bacteria are going to stand on the nose, and they stand on the skin. So the skin prevents the bacteria from entering the body. If you look at your arm, hopefully you have skin. If you didn't have any skin, the bacteria would go onto the meat of the arm, the flesh, and then they go to the blood vessels and the nerves and the lymphatic system, and they would infect your body very easily. So when you have individuals who have been in car accidents and you notice that part of their skin is cut, well, guess what? Bacteria are going to enter the cuts and they're going to infect the body. So the skin is part of the barrier system, which is part of the non-specific system. If we notice in the nose, we also have hair. And let's say that we are dealing with something called TB, tuberculosis, and tuberculosis is a bacteria, and it's airborne. So when the tuberculosis goes into the hair, hopefully the hair traps the tuberculosis, and the tuberculosis can no longer enter the nasal cavity and go into the respiratory system. But there's also mucus, and right here we're going to have some mucus secretion. We're going to make this blue, and hopefully the tuberculosis stays in the mucus, and then we sneeze it out. Or we can get that mucus, make a ball, and swallow it and put that mucus into the gastric acid. So part of the innate or the non-specific immunity is also gastric acid. Whenever we have a cold, we might have different type of pathogens, but we don't care about the pathogen. We just know that we have something bad. And we're going to make a lot of mucus, we're going to swallow that mucus, and we're going to put that mucus into the gastric acid. So that mucus has a lot of pathogens, but we don't care about what specific pathogen it is. Maybe it's the flu, maybe it's tuberculosis. We don't really know. However, we're going to put that into mucus, we're going to swallow that mucus and digest it. The gastric acid hopefully kills a lot of the bacteria, and we don't care about what bacteria it is. That's why it's called non-specific. Also, it doesn't have to be a pathogen. Hair can prevent dust and pollen from entering the respiratory system. The mucus can also prevent dust or pollen from entering the respiratory system. That is non-specific. It doesn't have to be just pathogens. It could be items like pollen or dust. Inside the body, we have internal defenses, and these internal defenses are part of the innate immunity. And I like to think of the innate immunity as like you're just relaxing, watching TV at home, and suddenly the door is broken into and somebody enters the house. Well, you don't know who this person is. You don't know their specific name. But what you do know is that they are an intruder. They are a pathogen, and they have to leave the body. They have to leave the house. So you and your roommates have to kick them out. Who are your roommates? Well, first you have the mast cells, and the mast cells, they are little cells that have a lot, and I mean a lot of these granules. And whenever they explode, we're gonna make something that explodes, they release a bunch of granules. And these granules have something called histamine. And histamine is going to increase the inflammatory response. And it also increases vasodilation. A good example of when a mast cell will explode is when you have allergies. So these guys, the mast cells, these guys are typically for allergies, but they don't have to be. But a good example are allergies. So when you're running in the park and you have all that pollen, your face starts getting hot, your eyes start running, you start sneezing, there's more mucus buildup, and you just feel terrible. You have sinus pressure. Well, that's because the mast cells exploded and they release a lot of histamine. And histamine causes vasodilation. The blood vessels start opening up and there's more blood flow to that area. That's why you have redness in your face. And then you also have inflammation. Inflammation, all that does is that it's going to increase the heat. So that blood is more pronounced in that area, and that blood has heat. So since there's more blood in that area, there is more heat because the blood brings heat from the inner body outwards. And the reason why we bring heat is because sometimes viruses and pathogens, they don't like the heat. They actually die. In this case, viruses don't die, but they just don't like the heat. They could probably denature. They probably break apart in the heat. And so if we increase the heat, we can kill some of those pathogens. And that's what the mast cells do. Notice that it doesn't matter if we're dealing with pollen. It doesn't matter if we're dealing with pathogens. Because really, these guys right here are not natural to the body. And so we will classify them as foreign, if I can spell that, foreign. These are foreign. We don't care about their specific names. We just know, hey, you don't belong in the body. I'm going to do a non-specific response. I'm going to explode all my histamine, and hopefully that kicks you out. Next up is the natural killer cell. And we're really going to simplify the natural killer cell. When we're talking about the innate immunity, let's have a cell. We're going to call it natural killer. And then we're going to have these little buttons on the membrane. So we have the little buttons right there. And these are the receptors. Whenever we have any foreign item, they have little receptors as well. And these little receptors, we're going to do this. And whenever the natural killer cell joins with the foreign item right there, we're going to have some receptors match. So these guys will match right here, and then these receptors also touch. And whenever we have the natural killer cell touching the other foreign item, the natural killer cell is going to release its enzymes. It's going to release some enzymes. And these enzymes go and they start breaking holes into the other cell. So what are the enzymes that the natural killer cell releases? Well, it releases something called perforins, and it also releases something called granozymes. Perforins are going to be making the holes in the membrane, while the granozymes are going to enter the pores that we make, the little holes, and they're going to start breaking down the membrane. So for instance, let's say that this guy right here is bacteria. The perforins are going to be released by the natural killer cell. They're going to be making holes in the bacterial membrane. And then the granozymes, also released by the natural killer cells, are going to go inside the bacteria and they start digesting, not really digesting, but they start breaking down the bacteria. So the natural killer cell makes perforins and also makes granozymes. And the reason why we say it's non-specific is because it doesn't matter if this is called bacteria A or bacteria B. It doesn't matter because all these bacteria express some sort of protein that the natural killer cell recognizes as foreign. And therefore it's going to have this reaction. Natural killer cells can also kill friendly body cells that are infected with viruses or bacteria or parasites, and they can even kill tumor cells. Let's say that this is a tumor cell. You can also imagine a virus inside of it or a pathogen that is taking over the target cell. So normally target cells will make proteins, and those proteins are going to make different responses. If we have a virus, this virus is going to insert itself into the DNA, and now instead of making friendly proteins, we now make viral proteins, and these viral proteins are going to explode outside of the cell and infect nearby cells. If we have cancer, then we have the uncontrolled replication of proteins and we keep doing cell division, and that's really bad for the body. So the target cell has a way of telling the natural killer cell, hey, I'm infected, I need to be eliminated from the body. Okay, well, how do we do that? We create something called FAS. FAS, this is a little receptor on the top. And the natural killer cell is going to have something right here, and we're going to make this purple, and this is called a FAS ligand, FAS-L or FL. And the FAS ligand is going to bind to the FAS on the target cell. The FAS is only sent out into the membrane when there's something wrong with the cell, and the cell is telling the natural killer to say, hey, I'm infected, I need to be eliminated. When the natural killer cell binds to the FAS, it will trigger a response inside the target cell that explodes it. So this is called apop, because we're popping it, apoptosis. So when you think of FAS and FAS-L, FAS ligand, you should think of apoptosis. So essentially the natural killer cell is going to bind to the FAS, and it's going to initiate self-destruction for that target cell. So eventually it starts doing its little signals. They have the signals congregating here, and eventually the target cell kind of dies. So that triggers apoptosis. And also, it's not granyzymes, but rather it is granzymes. So that's an honest mistake. It's granzymes, like granules, and they're enzymes, so granzymes. And then you also have the phagocytes. Specifically, you have monocytes, and monocytes can be macrophages. So we have macrophages. And monocytes can also become dendritic cells right there. And of course, we have the neutrophils and the macrophages. So when we get cut, the first people to enter the scene of harm are the monocytes, the neutrophils, and the macrophages. There's also mast cells and natural killer cells. Basophils, they kind of do the same thing as mast cells, but they are expressed less in the body. So there's not a lot of basophils inside the body. There's only like 1% floating around in the blood. We also have something called the complement system. And the complement system, that's kind of part of the innate immunity and also part of the adaptive immunity. But essentially, the complement system is how the body gets rid of the pathogens. So we have this little pathogen. We're going to mark it with something. And then some proteins are going to start building on the surface of the pathogen. And essentially, over time, that pathogen is going to start having holes because these little red proteins created a tunnel right there. And now water from the body can enter the pathogen, and this pathogen swells up like a balloon and it explodes. So that's called fixing complement. And when we use it as part of the adaptive immunity, we're going to be using these antibodies. When we use it as part of the innate immunity, there's some proteins already floating in the blood. And these proteins just kind of bind non-specifically to any type of bacteria or pathogen or parasite, whatever it is. And hopefully it starts breaking down that pathogen and it kills it. So that's part of the innate immunity. But for the adaptive immunity, in order to make these little holes inside of the pathogen, we're going to need those antibodies. And these antibodies are part of the B cells, specifically the plasma cells. And we're going to talk about complement systems further in the future in greater detail. Something that is really important to the innate immune response is called interferon. So these interferons are going to interfere with the pathogen's ability to harm the body. So let's say that this cell is infected with a virus, and this cell is now making viral proteins. The cell realizes, hey, I'm infected, I'm making something bad, I don't want to do that. So I'm going to send my neighbors a warning. I'm going to make interferons, and these interferons are going to go to my neighbors, and they're going to say, we got to decrease the amount of protein synthesis that we're making. So we got to decrease the amount of proteins that we make. So in case you're infected with a virus, the virus can no longer make any proteins. And if it does make any proteins, it's going to be a reduced amount. If we have cells that are infected, The interferons are going to target that cell, and it sends a signal for this cell to self-destruct. It's going to induce apoptosis. The interferon is also going to activate immune cells, for instance, macrophages. It can even activate T cells, maybe even B cells. But you should just know that interferons are part of the innate immune response. They decrease protein synthesis. They allow the infected cells to undergo self-destruction, also known as apoptosis, and they recruit macrophages, T cells, and B cells. Next, we have the adaptive immunity. So the first step was to do an innate response. We have a lot of bacteria, we have a lot of pathogens inside the body. I don't care about their names right now, I just know I gotta get rid of them. That's non-specific, that is innate. The adaptive immunity, it doesn't use the skin, it doesn't use the hair or barriers, but now it uses something called B cells and T cells. And these guys are part of the adaptive immune response. And this is specific. So in the innate immune response, we didn't care about the names of the bacteria, we didn't care about the names of the virus, but in the adaptive immune response, we do care. We can say, oh, we actually have an E. coli infection, we have a tuberculosis infection, we have the mumps or rubella. Maybe I have somebody in my body that can get rid of the mumps. Maybe I have someone who can get rid of the tuberculosis. Let's look for a specific person to get rid of this tuberculosis. It is more specific and adaptive. This is actually a slow response. And this innate is a fast response. So first we're going to do something that's fast, and that buys the body time to start the adaptive response. Part of the adaptive response are the T cells, the B cells, and the antigen presenting cells. Specifically, these are going to be the professional, let's write that, antigen presenting cells. So what are these guys anyways? Well, these guys right here, they are the dendritic cells. That's like the main person. We also have the macrophages. And we can also have the B cells, but that's not really common. The main person that does the presentation of antigens are the dendritic cells. This is a professional antigen presenting cell. For instance, you can have something like a macrophage, a B cell, or in this example, we're dealing with an dendritic cell. And you can look at the dendritic cell and see that this dendritic cell has a lot of arms. And all these arms can touch a bacteria, they can touch a virus, they can touch a parasite, and they will phagocyze these pathogens. For instance, here we have a pathogen touching one of the arms, and now it's being swallowed. When it enters the antigen presenting cell, it binds into a vesicle, and a vesicle is really just a transport. It's like a taxi for the pathogen. So the pathogen enters the vesicle, and now you have a lysosome. This lysosome binds to the vesicle, and it releases enzymes. These enzymes start breaking down the pathogen, and they create something called antigens. Antigens, all they are, antigens, it's the N, all that is that it's a protein. And you can think of it as a name tag. So for instance, we don't know what this pathogen is. Do you know the name? I don't. But if we swallow it and we start digesting it, we will make a name tag. And let's say that we are dealing with something called E. coli. So now this antigen has the name tag. This antigen says, hey, it's E. coli. Okay, so we have this name tag. What do? We're going to put this name tag, and then we're going to attach it to a piece of paper. What is this piece of paper called? Well, remember, this piece of paper is the major histocompatibility complex. It's the major histocompatibility complex. So now this antigen is going to go onto this piece of paper, And it's going to say, hey, we're dealing with E. coli. E. coli. So we have the piece of paper, we have the name on it, that's the antigen on the major histocompatibility complex. And then we got to present this paper to somebody. Who do we present this to? Well, we put on one of our arms, we're going to hand this piece of paper with a name tag on it, and we present it to a T cell. That T cell is going to look at that piece of paper, is going to see the specific name of the pathogen, and it says, hey, we're dealing with E. coli. Do I have anybody that can deal with E. coli? Oh, yeah, this special type of B cell can make antibodies for E. coli. Let's call him over. He's specific to E. coli. So this T cell can release little signals, and these signals right here, they are called cytokines. Cytokines. What cytokines do is that they are messages for other immune cells. So when the T cell receives this piece of paper with E. coli, it's going to release cytokines. And it says, hey, there's a message right here. It says, hey, I need a B cell that can kill E. coli. And now a B cell is called over, the B cell, and specifically he's for E. coli. So it says, oh, you called me over? For sure, I can get rid of this E. coli. Let me go find more E. coli. So this cytokine communicated with this E. coli specific B cell, and now an immune response can be performed. And now you might be noticing that there are two ways that this name tag is made. Let's call this pathway 1, and let's call this other one pathway 2. Pathway 2 is specific for extracellular pathogens. And pathway 2 is going to create something called the major histocompatibility 2 complex. So this piece of paper specifically is the major histocompatibility 2 complex. What about the other pathway? Well, the other pathway, let's call that pathway 1, that's specific for intracellular pathogens. So what does that mean? Well, that must mean that we have a cell, and this pathogen is going to be affecting the inside of the cell. So for instance, viruses are always going to be intracellular. You can have parasites that live inside the cell. So here's a parasite, it's like a little worm or something. It's living inside of the cell. You can also have bacteria that live inside the cell. So let's make a little bacteria that lives inside the cell. These are intracellular pathogens. They enter the body and then they make a home inside the cells. And so we need a specific type of piece of paper. So the piece of paper that we use is going to be this nice turquoise color, this nice teal color, and this is going to be called the major histocompatibility 1 complex. When we have a pink piece of paper, that is for extracellular pathogens. Those are major histocompatibility 2 complexes. But now we have to ask ourselves, who is reading these specific pieces of paper? Why do we have major histocompatibility 1 and major histocompatibility 2? Well, major histocompatibility 1 is only going to be read by CD8 cells, also known as cytotoxic T cells. And the major histocompatibility 2 complexes are going to be read by CD4 cells, also known as T helper cells. Here is a great example of what we're discussing. We have a professional antigen presenting cell, also known as a dendritic cell, and this dendritic cell is going to be presenting an antigen to the T helper cell. And the antigen is going to be placed on the major histocompatibility 2 complex. Since we're using the major histocompatibility 2 complex, that tells us that the pathogen is extracellular. That means that it doesn't have to live inside the cell to infect us. So we automatically know that we're not dealing with viruses. Now the T helper cells are going to be reading the name tag, the antigen, on the MHC2 complex, and the T helper cells are going to release cytokines or cytokines. These cytokines are going to be messages that talk to other immune cells. For instance, these cytokines can talk to CD8 cells or cytotoxic cells. They can also talk to B cells or macrophages or whatever immune cells they want to. They can increase the immune response, they can even decrease the immune response. And specifically, there are two types of helper cells. There are helper 1 cells and helper 2 cells. Th1 cells or T helper 1 cells are going to release cytokines that will communicate with T cells and other immune cells such as macrophages. They can also increase B cells, but not really. It's very weak. If you want to increase B cell, you're going to be using T helper 2 cells. T helper 2 cells, they're really going to be communicates with B cells. We can choose to increase their activity or we can choose to decrease their activity. But now we have to ask ourselves a question: how does the body know when to increase immune response and when to decrease immune response? Well, the T helper 1 cells and the T helper 2 cells are going to release cytokines, but the T helper 1 cytokines are a lot different than the cytokines from the T helper 2. Specifically, we have something called the interferon. We have the interferon gamma cytokines, and we also have the transformative growth factor beta. And all you got to know for right now is that interferon gamma is going to be the gas pedal. So if we're comparing the immune system to a car and we want to go faster, we want to increase immune response, this is going to be the gas pedal. While the transformative growth factor beta is going to be the brakes. And transformative growth factor, it's really complicated. You can write a whole paper on transformative growth factor because it's really related to tumor suppressing. We use transformative growth factor beta to decrease the amount of cell reproduction. Sometimes transformative growth factor beta can increase cell production, but it's really complicated, and for right now we're just saying that TGF-B will decrease immune responses and cell division, while interferon gamma is going to increase immune response and inflammation and will help us get rid of pathogens. So this guy right here is going to be pro-inflammatory. Right here, interferon gamma. And these are going to be secreted by T helper 1 cells. And they are special types of cytokines. But what about T helper 2 cells? What kind of cytokines do they make? Well, we make something called an interleukin. What is an interleukin? An interleukin means between white blood cells. So this is a message that is going to be used between white blood cells or between immune cells. So these interleukins are written as IL. So we have IL, and specifically for Th2 cells, we make IL-4, we make IL-6, and we make IL-10. And all you gotta know is that interleukin 4, 6, and 10 are made by T helper 2 cells, and they are going to increase the B cell activity. And also, the interleukin 10 is going to increase inflammation. So the pro-inflammatory characteristic of interleukin 10 is going to help increase the immune response, and that's going to help kill the pathogens. And so as a recap, when we're looking at CD4 cells, another way that we can name them are called helper cells, helper T cells. And these helper T cells have helper T cell 1 and helper T cell 2. Helper T cell 2 is really going to help out the B cells. And then the helper 1 cells, that's going to dictate the overall immune response. Do we want more immune response or do we want less immune response? If we want more immune response, we're going to be secreting interferon gamma. If you want less immune response, we're going to be secreting transformative growth factor beta. B is for brakes. Put the brakes, and now the car slows down. And that is all dictated by CD4. And again, CD4 is going to be using what? It's going to be using the major histocompatibility complex, and that is the second one. So the major histocompatibility 2 complex. And that's only for extracellular pathogens. We just finished talking about the T helper 1 cells, and we realized that T helper 1 and T helper 2 cells, they don't kill pathogens. They just increase or decrease the immune response, and they also communicate with B cells. If you want to kill pathogens, we have to use something called a cytotoxic T cell. And cytotoxic T cells, those are CD8 cells. And these CD8 cells are going to be what? They're going to be using the major histocompatibility 1 complex. And they are going to be intracellular pathogens, for instance, viruses, or maybe there's a bacteria right here, so this is a friendly cell, and now we have some sort of bacteria that's living here, and it's infecting the cell. So because we are inside the cell, it's a lot harder for the body to get rid of this pathogen. We're going to be using the cytotoxic T cell because the cytotoxic T cell can touch this infected cell and cause it to explode via apoptosis. How do we do that? Let's say that we have a dendritic cell. This dendritic cell is going to take this virus, and we don't know what kind of virus it is yet, but if I break down this virus and I separate the antigens, the proteins from the virus, I can have a name tag. So now that I have this antigen, let's say that we're dealing with the mumps, and the mumps virus is inside the body. So now I put this antigen name tag onto a major histocompatibility 1 complex. Now we have the CD8 cell. The CD8 cell is naive because it doesn't know any pathogens. It's just not doing anything. But when it reads the name tag on the major histocompatibility 1 complex, it says, hey, we're dealing with the mumps. And you know what? I have to get rid of cells that are already infected with the mumps. And those cells are going to have the name tag mumps or antigens. So these cells are going to have the antigens because they are infected with the virus. And in Zig's example, it's the mumps. So let's say that we have the mumps right here, right there, and we can see it right here. So this cell is now making viral proteins. It's infected, and it will take one of the antigens of the mumps and it will present it on the major histocompatibility 1. The CD8 cell is already activated and it's specific for this virus. So now it's looking for this name tag. Oh, I see a cell that has the mumps name tag. I'm going to lock into it, and when I touch this receptor, I'm going to start releasing my enzymes, specifically perforins, which are going to make little pores inside the cells. And then when I have holes inside the cell, I'm going to release granzymes. These granzymes are going to go into the cell and they're going to start degrading the cell. So now we have these large tears, and this friendly cell is destroyed. We don't want the cell to create more mumps virus, and so we had to destroy it as a sacrifice. If I allowed it to live, it will create those viral proteins and infect the body even more. So I had to get rid of the cell for the greater cause of the body. And so to recap, we take this virus, we don't know the virus name, we're going to start digesting it, we make an antigen, we present that antigen on the MHC1 complex. The MHC1 complex is only read by CD8 T cells. The CD8 T cells are now specific to this virus. In this example, it is the mumps. It will start looking for cells that are infected. Cells that are infected with this specific virus or pathogen are going to have this antigen on the surface. The CD8 cell is going to bind to this antigen on the antigen receptor, and then is going to start releasing the enzymes that ultimately kill those viruses. Or not viruses, but rather the cells that are infected with the viruses. You can't kill a virus because it is not alive. However, you can kill the cell that is infected with the virus. And this brings us to another great point. Let's say that we have these two cells, and one cell is infected with the mumps virus, and the other cell is infected with the rubella virus. And then let's say that we have a CD8 cytotoxic T cell. Well, this cytotoxic T cell is going to be specific towards the mumps in this example. Therefore, it's going to have a receptor that matches this receptor. And when they bind, the infected cell is going to be destroyed. However, if we take this mumps-specific CD8 cell, it is not going to have any effect on this rubella-infected cell. They can try to connect, but nothing is going to happen because this cytotoxic T cell is only specific towards the mumps antigen. So we need another cytotoxic T cell, and it's going to be specific towards rubella. And so now this cytotoxic T cell can bind to the antigen for this cell, and it can destroy this rubella-specific cell. And what I'm trying to say is you cannot have the cytotoxic T cell destroying all types of infected cell. That's not true. It can only attack specific cells. If it's mumps cytotoxic, then it has to be a cell infected with the mumps. It cannot be a cell infected with rubella or the measles. And fun fact, the measles are really just rubella. So it's kind of like the same thing. Fun fact, T cells, specifically cytotoxic T cells, so we're dealing with CD8 cells, they can also have the Fas ligand. A good example of this is when you're dealing with tumor cells. So these tumor cells recognize that something is wrong with them, and they will express these little molecules on the surface called Fas, F-A-S. When they bind to the Fas receptor on the cytotoxic T cell, the cytotoxic T cell is going to send a signal, and the signal is going to cause apoptosis inside the target cell, and this causes an explosion inside the target cell. So this helps to reduce the amount of tumor cells we have in the body. Another cell that can do this are the NK cells, the natural killer cells. So T lymphocytes, the cytotoxic cells, and the natural killer cells can use the Fas ligand to help induce apoptosis in tumor cells. Here we have another example. Let's say that we have some viral material and we don't know what this virus is. But this professional antigen presenting cell takes this virus material and is going to create an antigen. And when we have the antigen, we know specifically what this virus is. And let's say that it is rubella, the measles. Now we're going to take this antigen and put it on the major histocompatibility 1 complex, and the CD8 cells, the cytotoxic T cells, are going to read this major histocompatibility 1 complex with the rubella antigen. And now cells that have this rubella antigen on them are going to be destroyed by the T cells. However, after we finish destroying these cells, the body starts making memory T cells. So what are memory cytotoxic T cells? That's the body's way of remembering the antigen. Well, why do we want to remember the antigen? Well, we have two different immune responses. We have something called the primary immune response. And then we also have something called the secondary. We have the secondary immune response. The primary immune response happens when we don't have a prior experience with a virus. In this case, we're dealing with viruses since we're dealing with CD8 cells. So let's say that for some reason I was never given the rubella vaccine, and somehow I contract rubella. So now I'm really sick, I have symptoms, I have a fever, and I'm sick. Everything hurts. The body is trying to fight the infected cells. That is where you have symptoms. When they fight the infected cells, we're trying to build an immunity to that sickness. Eventually, I start feeling better, and now I no longer feel sick. I no longer have symptoms. We won that battle, but the body makes memory cytotoxic T cells. So if I get sick with the virus again, with the rubella virus, the body will quickly act. It says, hey, we have a lot of memory cells. If one of these little cells right here present with the rubella receptor, we can kill this cell before it can explode and create more rubella viruses. So on the second immune response, this is actually faster. This is faster. And sometimes I don't even get sick. Not sick. And there are no symptoms. So the secondary immune response is when you contract the virus again, and your body is a lot quicker in fighting those cells that had the virus because it has previous knowledge on how it dealt with it in the past. It has those memory cytotoxic T cells. And really, you can think of it in the B cells. So this is T cell specific, but we also see the same thing in B cells. And this gives us a nice leeway into the discussion of B cells. But before I talk about B cells, I want to talk about one last type of T cell. So we talked about the T helper cells and how there's T helper 1 and T helper 2. And we also talked about how there's cytotoxic T cell, which is CD8. So this is going to be CD8. And these guys right here are going to be CD4. But there's there's another type of T cell, and those are called T regulatory cells, TREG, or TREG. And these guys are T regulatory cells, and we just discovered them a couple of years ago, so we're still doing research on the regulatory T cells. However, we do know that they help suppress the immune system. And so if we keep creating these clones of the cytotoxic T cells, and we keep creating clones and more clones and more clones, that can lead to an autoimmune disease. And we don't want too much of a good thing, right? And so the regulatory T cells will suppress this action. They will limit how many memory T cells we have. And they will limit the activity of the cytotoxic T cells, and it helps the body. So we don't want too much of a good thing. And how do we do this? Well, it's going to release something called CD25, and also we have FOXP3. So FOXP3 and CD25 are going to be reacting with the T cells, and they help regulate their activities. They help to suppress the activity. So it's kind of like the brake pedal of the immune response for the T cells. But now, since we finished talking about the T cells and how it helps to destroy pathogens, let's talk about the counterpart, B cells. Do you remember when we were talking about T cells, and the T cells were immature? They were actually thymocytes. And the thymocytes were learning at Thymus University on how to become CD4 cells and CD8 cells. Well, the same thing is true for B cells. Instead of going into the thymus, they stay in the bone marrow for their education. So they go to Bone Marrow University. And at Bone Marrow University, they learn not to attack the body itself. They don't want to bind to self-antigens. So what is this process called? This process is called central tolerance. And we've already seen central tolerance for the T cells. The central unit for the T cells was the thymus. The central unit for the B cells are the bone marrow inside the bone. So the bone marrow is the central region. This is why we call central tolerance. Here we have the B cell. It's young and it's learning how to react to the body. And it bound to a self-antigen. It is not supposed to do that. If we have a B cell that binds to self-antigens, it will start hurting the body. And we don't want that to happen. So we have three options. The body can decide to edit the B cell, and it can edit the little receptors so that it doesn't bind to self-antigens anymore. And that's what happens here. We have a non-self-reactive B cell. It's not going to bind to the body anymore. Or we can induce apoptosis. So if we have a B cell that binds to the self-antigen, it can explode and it can induce apoptosis. And we no longer have that B cell. Or we can have the next option, and this is called anergy. Anergy. So if we have a B cell that binds to the self-antigen, we can make that B cell just non-reactive. It can no longer bind to anything. So no longer binds to anything. What does that mean? Well, it no longer binds to self-antigens, but if we have something like E. coli, and we have the E. coli antigen, the E. coli antigen is not going to bind to the anergic B cell. The anergic B cell is pretty much a dud. It just doesn't do anything. How does the body dictate whether to do apoptosis or whether to do anergy? Well, if the B cell really binds to the self-antigen, like it's super glued on and you can't yet get off, well, you have to delete it. You have to do apoptosis. However, if the B cell is kind of just pinching the little self-antigen and it can easily be moved away, it undergoes anergy. After the central tolerance, we now have the peripheral tolerance. The central tolerance occurred in the bone marrow for the B cell, but the peripheral tolerance occurs outside of the bone marrow. So it can happen in the lymph node or inside the lymph fluid. But essentially, this is where the B cell learns what a pathogen looks like. It reads a pathogenic antigen. So for instance, if we have an E. coli infection, this is a common bacteria, we have E. coli, the E. coli is going to be eaten by this antigen-presenting cell, the dendritic cell. This dendritic cell is going to put this antigen, we have this little antigen being made, and is going to be putting it on the major histocompatibility 2 complex. The major histocompatibility 2 complex is going to be read by the B cells, and it's also going to be read by T cells, specifically the CD4 T cells. Why do we need both the B cells and the T cells? Well, the B cells are really eager. They really want to do something quickly. They say, oh, I finally have a signal. It's E. coli. Oh, I want to do something. Let me do something really quickly. It's so eager that it needs a supervisor. It's kind of like getting a new kid on the job and you got to supervise what this person's about to do. So the T cell is going to give another signal to the B cell and is going to release little cytokines. And this is going to be via the helper 2 cells because that is for the B cells right there. So now the B cell received 2 types of signals. It received the signal from the antigen receptor, we're going to call that AR, and now it received a signal from the helper 2 cells. Now that we have both signals, the B cell can now create antibodies. So now the B cell can make antibodies. And the antibodies, we'll talk about them later, are going to help kill pathogens. So to recap, for the B cell to even start working, we're going to need 2 signals. And that is from the antigen receptor and also the CD4 T cell. Specifically, T helper 2 cells. T helper 2 cells. Let's say that we have these little cells, these little proteins floating inside the fluid, and these are self-antigens. And they're just floating around. If the B cell binds to one of these self-antigens, so we have a little receptor and they bind to these self-antigens, it's going to say, oh my gosh, I have something on my membrane, I have an antigen, I want to react to it, I want to mount an immune response to this antigen. But the T cell looks at it and it says, really? No, don't do that. That's a self-antigen. I'm not going to send you a signal. I'm not going to do any cytokines for you. And now the B cell is given another signal to explode. So this induces apoptosis. So the B cell is like a really little kid. It's so eager to do something. It really wants to help out, and it's going to bind to antigens. When it binds to pathogenic antigens, that's a good thing. The T cell is like a father. He's going to give a pat on the back to the B cell and says, hey, you can do it. Go make your antibodies. And now the B cell is a little kid and he's all happy. He's like, thanks, Dad, and he's able to do his job. However, sometimes the B cell is too eager and it says, oh my gosh, I have an antigen. Can I make antibodies to it? It's a self-antigen. And so the dad says, no, son, go to your room and go study some more. You need some more work. And now the B cell, unfortunately, undergoes apoptosis, or it's no longer able to function. So that is the peripheral tolerance. Here we have the antibodies. And when we have an immune response, the B cell is going to convert into a plasma cell. And the whole purpose of a plasma cell is to create those little antibodies. And specifically, they are towards the pathogen. So for instance, if we have mumps, we're going to make antibodies that are specific towards the mumps. If we have rubella virus, we're going to make antibodies that are specific towards rubella. If we have the E. coli bacteria, we're going to make antibodies that are specific to E. coli. If we have a parasite, we're going to make antibodies that are specific towards the species of parasites. And so antibodies are going to mark viruses, bacteria, parasites for elimination by the immune system, and they're very important for the immune system. There are 5 different types of antibodies. The first one is called IgG. And we have to ask ourselves, what does Ig mean? Well, Ig just means immunoglobulin. That's all that means. Immunoglobulins is another way you can call antibodies. So this is an immunoglobulin, and this is an immunoglobulin. So we have immunoglobulin G, we have immunoglobulin A, we have immunoglobulin D, E, and finally we have immunoglobulin M. And each immunoglobulin consists of heavy chains and light chains. There's also a constant region and a variable region. What is the constant region? Well, the constant region allows us to differentiate between immunoglobulin G and immunoglobulin A. So for instance, you might have immunoglobulin G right here, and it has a certain amino acids, and it's always going to have this the same strand of amino acids right here. But what if we change the amino acids? So instead of being dark blue, we're going to have this nice teal. Well, when we have a different strand of amino acids inside the heavy chain, we're going to have something called immunoglobulin A. So the heavy chains differentiate between the immunoglobulins. You might have alanine, and I'm making this up, alanine, valine, that codes for immunoglobulin G. You might have valine, and we have leucine. And that codes for immunoglobulin A. I'm not saying that it does, but in this example, I'm saying that the heavy chains are dependent on the amino acids that create them. Certain strands of amino acids will code for the immunoglobulin G, and certain amino acids are going to code for immunoglobulin A and D or E or M. And now we have to ask ourselves, what does the variable region do? Because the constant region, that tells us what we're going to be. We're going to be IgG, we're going to be IgD. But what do the variable regions tell us? That tells us what we are specific towards. So depending on the amino acids expressed here, we might be specific towards the mumps, or we might be specific towards E. coli, or we might be specific towards a certain type of parasite. And what I'm trying to say is, we can have something like this. We can have IgG for mumps, and then we can have IgG specific towards E. coli. So notice that these guys are both IgG, but this guy only attacks mumps, and this guy only goes to E. coli. And that is because there is a difference between the variable region. They both have the same constant region, which is IgG, but the variable region is different, and that allows us to target different pathogens, different bacteria, different viruses, different parasites. Now before I leave this slide, I want to clear the board and I want to point your direction towards this section. So this section right here is called the FC. That is really just fragment crystallizable. And what this does is that certain cells are going to bind to this FC binding region and they're going to react. So for instance, when we have IgE, this guy is for allergies. So let's say that you are allergic to shrimp. So we have a shrimp particle right here, and this shrimp particle is going to bind to this region, to the variable region of the IgE. And now this IgE particle right here for shrimp, we're going to call this IgE shrimp. It's going to go to another cell, and this cell is called a mast cell, and this cell has a lot of histamine inside. This mast cell is going to bind to the FC region of the IgE, and then it's going to explode. So it's like getting a grenade, pulling the pin and throwing it. The mast cell explodes and it releases histamine, and that causes an allergic reaction. Now your bronchi, your lungs, they start to constrict and it's hard to breathe. Your face is flushed because there's vasodilation. You feel hot because of that vasodilation. And so what I'm trying to say is that the FC binding region is going to bind to cells that have FC receptors. So this little guy has FC receptors. It's going to bind to the FC region of this IgE, and now it's going to induce a reaction. So this doesn't only occur with IgE. IgG also has an FC binding region. When we look at the FC binding region of the IgG, this guy is going to bind to macrophages. So here we have a pathogen, and this pathogen has a lot of antibodies on it, right there. And notice that the little tips right here, these little tips, are the FC binding regions. When we surround the pathogen with antibodies, we make it harder for the pathogen to move. We also make it harder for the pathogen to infect other cells. And this whole process is called opsonization. Opsonization. That's when we surround the pathogen with antibodies. So that is when the pathogen has antibodies on it. Now going back to the FC binding region, the FC binding region of the IgG, that's going to attract macrophages. So not only are the antibodies going to opsonize the pathogen, they prevent it from entering cells, they prevent it from infecting cells, and they slow down the pathogen so that the macrophages are able to phagocyze the pathogen. So that is opsonization. And so we learned that the FC binding region for IgG communicates with macrophages, and it highlights the pathogen. It slows down the pathogen, and now it makes it easier for the macrophage to capture the pathogen and to swallow it and destroy it. And that's really crucial for bacteria or parasites. And here are the 5 different types of immunoglobulins that the plasma cells create during an immune response. We have IgM, IgG, IgA, IgE, and IgD. One important thing that you have to remember is that we're going to be dealing with the FC binding regions. So the immunoglobulin has the FC binding region right there. The IgE is also going to have an FC binding region. And you're probably saying, oh, the IgD also has an FC binding region. Well, not really. I mean, it does have a binding region right there, but this guy is always going to be bound to cells. Specifically, it's going to be bound to T cells and also B cells. And you're probably thinking, well, what do you mean? If we go back into the beginning of the lecture, you will recall that you have your T cells, and you also have your B cells, and these T cells are going to have little receptors. It might have a CD8 receptor, it might have a CD4 receptor, right? So you can either have a CD8 or CD4. For the B cell, we also have receptors. That's how we read the antigens presented to us by the dendritic cells. But these guys right here, these are actually IgD. They are IgD. So the IgD is going to be making the receptors for immune cells. And that's why we don't really talk about IgD. We don't really test for it in the blood. But what we do test for is IgG, IgM, IgA, and IgE. When we look at the Fc binding receptor, that goes to macrophages, and that helps in opsonization. For the Fc binding region for IgE, that goes into mast cells, and that triggers an allergic reaction. They can also trigger anaphylaxis. So anaphylaxis is a severe allergic reaction, and it's an emergency for people. So for instance, if somebody eats shrimp accidentally, they have an anaphylactic reaction, they can't breathe, and they need immediate emergency attention. So that's because the Fc binding region of the IgE has reacted with a food particle, and it touched a mast cell, and the mast cell released all its histamine. When you think of IgA, you should be thinking about babies, because IgA is an antibody. It's an antibody that can be secreted in the breast milk. So when a baby is born, it's very sensitive to the outside world. It has a weak immune system. When the mother breastfeeds the baby for the first time, the immunoglobulin A goes into the breast milk and it enters the baby's digestive system. So now the baby has a stronger immune system thanks to the mom breastfeeding the baby. An easy way to remember IgA is to say, "Aww," because when you see a baby, you go, "Aww, it's so cute," you know? So IgA, IgA. Not only that, but IgA can be found inside the sweat glands. So it could be found in sweat, saliva, and the skin. So when we're thinking about the skin, which is a part of the innate immune system, if a bacteria lands on the skin, sometimes there's going to be IgA, and that IgA antibody on the skin is going to prevent the bacteria from moving any further. And it's really good. So when you think of IgA, you should be thinking about fluids, because it's found in sweat, saliva, it's found on your skin, it's found inside breast milk, it's also found in the tears. When you're crying, the tears have IgA antibodies in them. Here we have the IgM, and the IgM is the largest antibody. IgM is produced the most during a primary immune response. And you're probably wondering, hey, we also had immune responses for T cells. We have the same thing for B cells. In fact, during a primary immune response, that's the first time I've ever encountered a pathogen. So let's say that I was at a salad bar, I was eating salad, and some of the salad had E. coli. Now I have symptoms. Now I feel sick, I'm vomiting, I have a fever, I just don't feel good. So the first type of antibodies that we're making is IgM. IgM is like the first line of defense for brand new pathogens. So now the body is using IgM to get rid of this E. coli bacteria. So this is an extracellular pathogen. It also works for parasites. So really, antibodies are going to be targeting things outside of the cell. And you'll see why in a few minutes. But notice that the IgM has so many antibodies pitted together. Well, that's great, because here we have an E. coli bacteria, we have another E. coli bacteria, and another, and another, and another. So this IgM can take care of 5 different E. coli bacteria. So the reason why we produce so much IgM is because we want to get rid of as much pathogens as possible. IgM can bind to 5 pathogens at once, as long as they're the same type of pathogen. You're not going to have You're not going to have E. coli and then Salmonella right there. That doesn't make any sense. They're all going to be E. coli in this example. IgM is a little special because IgM can do something called class switching. And what class switching does is that IgM eventually will convert itself into IgG. We can change our classes, but we don't change what we are specific towards. So we might have IgM for E. coli, but if I class switch, I now become IgG, but I'm still specific towards E. coli. And I don't have to be IgG, I can be IgA, I can be IgE. So it really depends on what I want to become. So IgM can become this guy, it can become IgA, and it can be IgE, depending on what the immune response is. And here we just have a picture that shows that during the primary immune response, we have a low concentration of antibodies. So we have a low concentration of, let's say, IgG. However, during the secondary exposure, if we're infected with E. coli again, we have a faster secondary immune response. And this secondary immune response is going to be stronger, it's going to be faster, and I'm not sick. Sometimes I don't even know that I'm sick. You can eat something, it has E. coli, but your body defeats the E. coli very, very quickly, and now you don't even feel sick. You didn't even know you were sick in the first place. But that's the secondary immune response. The reason why we have a secondary immune response is because similarly to the T cell, memories, to the memory T cells, we also have something called memory B cells. So we have B cells right there. So we have memory B cells, and let's look at them and how they function. Let's say that we have a dendritic cell, and this dendritic cell is going to take in a pathogen and it's going to break down this pathogen. Now it's going to secrete this pathogen right here on its surface, and then it's going to present it to the B cell. The B cell is going to take in this antigen and it's going to become partially activated. It still needs help from this little T cell right here, specifically the T helper cell. And if you want to be more specific, T helper 2 cells right there. So it's going to send these little cytokines onto the B cell. It's going to be activated, and now that it's activated, it's going to release, it's going to become either a plasma cell or a memory cell. So plasma cells are like the machine guns of the immune response. They're going to be shooting rapidly these antibodies. So for instance, we can create IgG, or we can create IgE, for example. And each plasma cell is really fast. It's about 2,000 antibodies per second. They're really quick. They have a large amount of protein synthesis. So they have a large amount of ribosomes inside. So they have ribosomes. Likewise, the B cell can also become a memory B cell. So after we defeated this pathogen, the B cell says, hey, I gotta remember how we defeated this pathogen. I gotta remember the antigen that is specific towards E. coli, or the antigen that is specific towards rubella, or the antigen that is specific towards mumps, whatever. When we get infected with the pathogen again, that's called a secondary response. And now these memory cells are going to create a lot of plasma cells, like more than 10. Definitely more than 10, okay? So we have a lot of these memory cells that are just wandering inside the body, and when they pick up an antigen, let's say that it's the E. coli antigen yet again, the memory cell is going to create a bunch of plasma cells, and these plasma cells are going to rapidly create IgG, and that's going to help defeat the pathogen yet again. And that's why the secondary response is so quick and it's so fast and so powerful, is because we have these memory cells. So whether you're talking about T cells or B cells, the memory cells are the reason why we have these secondary response. Now we're going to be talking about how B cells prevent further infection from pathogens. And it all starts with the antigen-presenting cell communicating with cytotoxic T cells and also B cells. You're probably wondering, hold on, cytotoxic T cells are CD8, and they use a major histocompatibility 1 complex. But the B cells, they are going to be using the major histocompatibility 2 complex. So how is this possible? How is it possible that the dendritic cell can communicate with cytotoxic T cells and also B cells? Well, they are professional. They can do both. That's why we call them professional antigen-presenting cells. On the surface of the dendritic cell, we have major histocompatibility 1 complexes. We also have major histocompatibility 2 complexes. When we're presenting antigens to the cytotoxic T cells, we're going to be presenting the antigen on MHC1. When we communicate with the B cells, we're going to be communicating with the major histocompatibility 2 complexes. Why do we want that? Let's say that we're working with the hepatitis B virus. And the hepatitis B virus has the ability to infect cells. So we're going to make this little pentagon right here. So first, this little virus is floating around the blood, and it is extracellular. But when it goes inside here, so we're going to have this little virus is going to infect the cell, and now we have the virus inside the cell. So this is the intracellular. And now it's going to start forcing the cell to create more hepatitis B viruses. We want the dendritic cell to take this virus and to recognize that it is foreign. When it takes in the virus, it breaks it down and it presents part of the little viruses, the viral antigens, on the major histocompatibility 1 complex. It now presents it to the cytotoxic T cells. Now the cytotoxic T cells are going to have the viral antigen right there, and they're going to be looking for it on the exterior portion of the friendly cells. And here in this example, this cell presented with the hepatitis antigen. And because it presented with the hepatitis antigen, the cytotoxic T cell is going to induce apoptosis on the cell, and it destroys this cell. It prevents the virus from replicating. And that is the intracellular response. On the extracellular response, that is outside of the cell, so we have extracellular, the virus is just floating around, waiting to bind to the surface of a cell. Again, we will take the same antigen, and instead of presenting it to the major histocompatibility 1 complex, we're going to present it to the major histocompatibility 2 complex. And what that allows us to do is that the CD4 T cells are going to recognize the viral antigen being presented by the dendritic cell, and so does the B cell. So now the B cell gets the viral antigen, and it says, okay, I got the first signal that we're dealing with hepatitis B, but I still need my secondary signal from the helper cell. Again, this is a TH helper cell. Specifically, since we're communicating with the B cell, is going to be a helper 2 cell. So now this helper 2 cell is going to release cytokines, and it says, yes, B cell, we're dealing with hepatitis B, and I need you to create more antibodies. So now the B cell becomes a plasma cell, and it starts shooting out antigens, and antibodies. And these antibodies are specific towards hepatitis B. So let's say that we're creating IgG antibodies. So now IgG antibodies are going to do what? They're going to surround the virus. So they surround hepatitis B, and now hepatitis B cannot bind to the surface of the cells. They cannot infiltrate the surface of the cells. And specifically for IgG, this allows the macrophages to say, hey, there's a virus right there. Thank you so much, IgG. And it's going to take it in, and it's going to phagocytize it, and it's going to start breaking down the virus using lysozymes and digestive enzymes. And so what we learned is that B cells attack invaders outside of the cell. And T cells, specifically cytotoxic CD8 T cells, attacked cells that are already infected by viruses. And one important thing to know is that all the cells in your body will express major histocompatibility 1 complexes, just in case if they are infected by a virus. However, not every cell expresses major histocompatibility 2 complexes. Those are often found inside immunological cells, such as B cells or T cells, not in the liver cell, for instance. Let's look at another example dealing with parasitic infections. So when we have a parasitic infection inside the small intestines, for example, macrophages and dendritic cells, they are going to phagocyze. They're going to take a little piece of the parasite, and they're going to create those little antigens. So first, we're going to start secreting these antigens on the MHC2 complex. So this is going to be on the MHC2 complex. And when we use the MHC2 complex, we are talking to the CD4 helper cells. The CD4 helper cells. In this example, we're going to be using the helper 1 cells. All you got to know about helper 1 T cells is that they're going to increase the immune response. Usually they increase the immune response. How do they do that? Well, they're going to be secreting something called interferon gamma. And interferon gamma is the gas pedal for the immune response. This is pro-inflammatory. So ultimately it's going to lead to inflammation. So hopefully we increase the temperature and the parasites don't like it and they leave the body. Usually doesn't happen, but the body tries it anyways. If we want to stop the immune system, or if we want to slow down the immune response, helper 1 cells are also going to secrete something called transformative growth factor, beta. And this is the brakes for the immune response. That's just kind of like a little side piece of information. So pretty much all that happened is that the dendritic cell took a piece of parasite, used it to make antigen, presented that antigen to the CD4 T cells, specifically the helper 1 T cell, and that increased inflammation. At the same time and in the same location, the small intestine in this example, the dendritic cell can take that piece of antigen and it will present it to the helper 2 cell. So again, this is another CD4 T cell, but specifically it is the helper 2 cell. And it takes that antigen and it's going to secrete cytokines. So it secretes little messages that go around the area. And some of these messages are going to be specific towards the B cell. Notice that the B cell also took in some of this antigen. So it got some of the parasite antigen and it says, okay, I realize that there is a parasite here. I got my first signal. Now I need a second signal from the helper 2 cell. Oh, here's the second signal, cytokines. And now I can actually become plasma cells. But what kind of cytokines are we dealing with? In this example, we're dealing with interleukins. And you don't have to remember the specific interleukins for now, but just realize that the famous ones are interleukin 4, 6, and 10. And for the most part, these interleukins 4, 6, and 10, they're going to increase the activity of the B cells. Now the B cells are going to create plasma cells, and these plasma cells are going to secrete antibodies IgE and IgG. When we think of IgE, you should be thinking about allergies, but you should also be thinking about parasites, a parasitic infection. And IgE is going to work very closely with eosinophils. If we have a large concentration of eosinophils in the body, we are most likely dealing with a parasitic infection, and you can start looking at anti-parasitic medication. Let's focus on IgE for a moment. IgE is being produced because we have a parasitic infection in this example. When IgE is produced, it's going to bind to the mast cell via the Fc binding region of the antibody. As soon as it touches the mast cell explodes and it releases all the granules. Specifically, it releases something called histamine. It also releases more messages. We'll talk about that in a few seconds. But histamine increases vasodilation, and it also increases inflammation. So this guy is also going to be increasing. So the inflammation increases. It also increases bronchoconstriction. So it makes it harder to breathe. Now, we don't only release histamines from mast cells. Mast cells also have these little messengers, and these little messengers, the cytokines, they're going to communicate with eosinophils, and they say, hey, there's some sort of infection going on, maybe there's a parasite, I need you to be activated. So these eosinophils say, okay, you got it, I'm going to be on the lookout for these pathogens. I just wish there is something that can stick to the pathogen and tell me, hey, this is a pathogen, let's do our jobs. Well, that's where the antibodies come in place. IgG is going to stick to the surface of the pathogens, and now the eosinophils are going to bind to the IgE. Not only that, but the pathogens, the parasites, they're moving really quickly, but when all the IgG antibodies stick to the pathogens, to the parasites, they're going to slow down and they can't move as fast. In addition to this, mast cells also produce something called leukotriene. So this is actually leukotriene C4. And all you got to know is that leukotriene, in general, always tries to recruit help. It always tries to recruit more immune cells. The specific leukotriene that we're dealing with is called C4 in this response. Well, parasitic responses in general. And all that does is that it's going to increase the production of mucus, and mucus is going to slow down the pathogens the parasites. When the parasites slow down, all the IgG antibodies bind on the surface of the pathogen. And now, since they bind to the pathogen, the eosinophils are going to bind to the IgG, and they will release their little granules. So now these have enzymes, and they start breaking down the pathogen. Sometimes we don't even need the eosinophils. Sometimes the little IgG will bind to this guy right here. They're going to start binding, whatever it looks like, right? And then a protein is going to come along and is going to start poking holes into the pathogen. And what is that called? That is called complement. And now we got to figure out how that works. This is called fixing complement. And fixing complement, that just means we're using antibodies or we're using proteins to create holes inside the pathogen. Let's say that in the previous example we were dealing with parasites. Well, this right here is the same parasite, and we shot the parasite with a lot of antibodies. And instead of using eosinophils, we're going to be using proteins to make those holes. The special type of protein that first begins is called a C1 protein. That is the C1 protein, and it's just floating around the blood. Eventually, it will be picked up by an antibody, and it begins a cascade. So this right here is part of a cascade. C1 is going to bind, or it's going to start the cascade, and C4 is going to bind to C1. C4 is then going to break into two pieces right here. So now we have two pieces of C4b from this one C4. After that, we're going to have C4b, and then we're going to have something called C2b. And the reason why we say C4b and C2b is because C stands for complement. So that's all it means. And B just stands for binding. So complement for binding is going to bind to complement to binding. So now we have C4b and C2b. They are going to stick together and they create something called the C3 convertase. So together they make something called a C3 convertase, and let's give that a shape, because if we don't give it a shape, we're going to be very confused. So we're just going to call this shape C3. And again, this little square, the C3, is on the surface of the pathogen. After that, here's our shape. The C3 is going to break into two pieces. So it's going to break into two pieces. The first one is called C3b. That is the binding portion. And then we also have 3a. You may be saying, oh, 3a, that's a small little piece of nothing. It's not important. It is important. The reason why it's important is because it's going to recruit help. It's going to calls macrophages. And it also increases the immune response. Now let's go back to the starting position. You remember how the C4 split in two? We only used one of those C4b's. There's another C4b right there. So what if we have another C2b? Well, that makes another C3 convertase. And so all we got to do is take this piece right here, is this C3b, and then we're going to move it all the way back here to the other C4b, C2b. So that's where a lot of people get confused. They're saying, why does it go back to the original step? Well, there was two C4b's to begin with. We used one to create the C3 convertase. That is going to split into two pieces, C3b and C3a. This C3b is going to go back to the other C4b, and that creates something called C4b, C2b, and then we're going to have C3b. That's all we did. And we call this guy the C5 convertase. And let's give it another shape. Let's make it a square and C5. What if we split the C5 convertase into two portions? What if we have C5b, C5 binding, and then we have C5a? Actually, just 5a. Well, 5a is going to do the same thing that 3a is doing. All this does is that it's going to recruit for help. This calls for more macrophages and more immune response. So it's very similar to 3a. Let's take C5b and let's put it on the surface of the parasite or the bacteria. Here we have C5b, and it joins with C6, C7, C8, and eventually C9. C9 has a shape that looks like a funnel. It looks like a cylinder. And when this occurs, when they all join together, they create a hole inside of the parasite or inside of the bacteria. And now water from the intercellular space is going to rush in, and it's going to expand this parasite, or it's going to expand this bacteria, and the pathogen can no longer expand and it ruptures. And so that is called fixing complement. That is the complement cascade. And this whole process right here, this little tube that we made, is called the membrane attack complex. But let's talk about the alternative pathway. Sometimes the pathogen, like a parasite or bacteria, they're just relaxing. They're not doing anything inside the body, and they're trying to be sneaky. However, the C3 protein will spontaneously attach itself to the surface of the pathogen. So there is no antibody. It's really sneaky. It hasn't been discovered yet. However, the C3 is going to bind to its surface because this pathogen doesn't have certain proteins. So for instance, a liver cell is not going to bind to the C3 protein because it has little proteins on its membrane that says, hey, I'm friendly, don't bind to me, don't hurt me. The pathogen doesn't have these proteins. And so the C3 is going to kind of touch the surface of the pathogen, and it realizes, hey, there are no inhibitory proteins on the surface, I'm going to bind to it. So now we have the binding of C3. C3 is going to break apart into C3 binding and C3A. Now that it realizes, hey, there's something weird here, let me call for reinforcements. C3A, or just 3A, since it's not going to be complementing anything, is going to call for macrophages. And that takes a little while for the macrophages to get there. But in the meantime, C3b is going to convert, is going to call in for more proteins. And it's a very complicated process, but essentially we're going to use factors P, B, and D. After they interact with C3b, we have a new protein. And this new protein is called protein B, and this protein B is going to be binding. So we call this BB. Protein B is going to bind to C3b. So C3b plus binding protein B is going to create something called C3 convertase. And we already know what happens with C3 convertase. So here's C3 yet again. And in this example, we just regenerated C3. So now C3 is going to split again. It's going to make C3b and just 3a. This C3b is going to bind to the pathogen again and is going to keep doing this process. And so on exams, you can say, hey, the alternative pathway regenerates what? It regenerates C3. But anyways, let's kind of step back for a minute. When C3b splits again, it's going to join with the C3 convertase. So we have C3b, protein B binding, and now we have another C3b. This guy is called C5 convertase. So let's give it another shape. C5, and now C5 goes to the binding portion of the pathogen, so the membrane. Here we have it, and C6 joins it, C7 joins it, C8 joins it, eventually C9, and it makes the membrane attack complex, and now water rushes into the pathogen and it explodes the pathogen. So a very quick recap: C3 binds to the membrane portion of the pathogen because there are no inhibitory proteins that prevent the C3 from attaching to it. C3 splits into two portions, C3 binding and 3A. 3A calls for more macrophages. C3b will be reacting with factors P, B, and D. Peanut butter is delicious. That's the factor. Now we have protein B binding. C3b, in addition to B binding, will create C3 convertase, which is this guy. So we just regenerated C3. We split into two portions yet again. So now we have C3b. When C3b attaches itself to the C3b and BB, it creates something called C5 convertase. That's all it is. C3b, BB, and C3b yet again. C5 convertase goes to the membrane, C6, C7, C8, C9, and then you have the destruction of the pathogen. Now let's talk about the lectin pathway. And you're probably groaning, oh, it's another pathway. It's easy, I promise. Sometimes we have bacteria that have certain sugars on this surface. And these sugars, these little carbohydrates, they are called mannose. And that bacteria has not been discovered yet by the immune system. So right now it's hiding. But there's a special type of protein called the mannose binding lectin, and it's a protein. And this protein is just randomly being, is just floating inside the blood. And this MBL will spontaneously grab on to this mannose carbohydrate on the bacterial cell wall. And it says, there's something wrong. I'm attached to something. I'm attached to mannose. And mannose is really present within pathogens. So now I'm going to start the cascade for complement. C4 is going to bind itself to the MBL. And now C4 is going to split. And we have C4b. C4b is going to connect to C2a. And then everything kind of follows the same pathway as the classical pathway. So the lectin pathway, the only difference between the lectin pathway and the classical pathway is that the lectin pathway doesn't need any antibodies. So no antibodies. And so if you know how the classical pathway works, you know how the alternative pathway works, and you know the lectin pathway. Pretty simple. All that's different is that we don't need antibodies for the lectin pathway. All we need is to bind to the carbohydrates, in this case mannose, and mannose is found on the cell walls of the bacterium. Then we have mannose binding lectin, which is a type of protein that binds to the mannose. After that, C4 attaches itself to the MBL, it splits, it attaches itself to C2a. This guy right here is called C3. C3 will split into C3b, and then C3b is going to bind itself to another C4b, C2b, this is B right here, and make C5. C5b is then going to bind to C6, C7, C8, C9, and create a hole inside the pathogen, and that's called the membrane attack protein. And really, those are the three pathways. Just for some reason, in this picture, they have C2a. That's really C2b. I think this is enough immunology for one day. And you should be good to understand the basics of immunology. So when an immunologist is speaking to you, for the most part, you should understand about 90% of what they're saying, at least the basic concepts. But thank you so much for just listening to me ramble and stutter and attempt to teach immunology. I mean, it's a lot. I can make a 3-hour video, maybe even 4-hour video on immunology. And of course, there's a lot of things that I left out, but hopefully you understand the basics of immunology. And I know your time is precious, so I'm going to leave you with this message. Thank you so much for listening to my lecture. You are amazing, you are awesome, and you are worth my efforts. You are worth the education that I give you. I love you so much, and you are amazing. If something feels hard to learn, it is hard. I struggled learning immunology. I'm not a master of this. I just took time studying. And I just want to share that education with you. How do you eat an elephant? One bite at a time. I love you, and take care. You're awesome. Bye.